Hey, how you doing? We did bacterial genetics. Okay, so starting off, this is the group. We got Sean, he's gonna be on mutant selections, myself on mutations, Sarai, she's got plasmids, Savannah's gonna do transmids, and Sam's gonna finish it off with mutant selection. Okay, so mutations are gonna happen through the central dogma process. This is when RNA goes to DNA, or DNA goes to RNA to proteins. The codes can get messed up, and then your proteins are going to end up messed up and different. That's known as the central dogma process. There's a quote from the textbook: "Spontaneous mutations occur routinely yet infrequently." That means that this doesn't happen a lot. If it happened a lot, we'd be very messed up because we're pretty much all proteins. And yeah, there's two types of mutations. There's spontaneous, which is the one that happens most often, like we see when we study. Then there's also induced ones, like radiation. If you've seen Chernobyl, there's weird creatures because that can be induced by stuff like radiation. Next. Okay, so codon sequences, I mentioned this before. That's kind of the code of amino acids, how they're going to be made. They're going to be made through these sugars. There's uracil, guanine, adenine, and C. So one small change in those can mess it up. So let's say one of these is changed, the whole protein structure is going to be different. There's a protein called RNA polymerase 2 that's going to read the DNA line like this, and it's going to change it into an RNA line. And then you have another protein called tRNA. It's going to go grab an amino acid and pair it up according to this. And if one codon is off, then the tRNA is going to grab the wrong protein, and then or amino acid, and then the whole protein, which is the polypeptide chain, is going to be messed up. There's three types of mutations. Silent is a change of one letter, but it still codes for the same amino acid. It doesn't do much in your body. You wouldn't never know that it happens. That's why it's called silent. It doesn't change the function. Then you have missense. Missense is going to change the codon and change the amino acid, but it's not going to really change the function. That happens sometimes. And then you have nonsense. That's where the letter gets changed, the amino acids get changed, and then the outcome is different. So then it kind of messes up your whole protein.
So the next topic that we're going to be covering is mutant selection. So what is mutant selection? Mutant selection is a way for scientists to make a better organism by causing mutations in the DNA. And you do this in various ways that we will cover in the upcoming slides. As you can see, these changes happen rarely, so you have to use a large population to achieve one result. Mutant selection is usually covered in plants or sometimes microorganisms. You can't do it on large animals or humans, of course, because you need a lot of population of that single organism, and plants produce a lot of seeds and offspring, so with microorganisms. Mostly used as plant breeding techniques to grow products faster and in large quantities. As you can see, this is an example of mutant selection. So they use a dose, so that could be radiation or something. If you have a little dose of radiation, you can see that the plant is going to grow. This can affect in a different way. It can grow faster or it can grow slower, but the more doses that you use on the plant, for example, it can become harmful. So as you can see, the more it increased, there were more harmful consequences, and then after a certain amount, it was damaged and died. Here's a picture explaining it a little bit better. So first we have our starting culture, and then mutant genesis, so a radiation or type of mutation is added to the offspring, and then at a molecular level, the DNA changes in some way or form, and then we have two different scenarios with that. So first we take our offspring, and we see which is the best product that we're trying to get from that, and we limit it down to 10%, and then see what's the best from there, and then we limit it down to another 10%, and then we see what is better from there. And then lastly, we take it from our 10% and choose probably the top 5 best offspring that was produced from that radioactive scenario. This can depend on what offspring that you're using with. And then We have another example right here. Sometimes scientists do radioactive on an offspring, but they don't really look for the best. They just look what changed in that big offspring. I got this all from a peer-reviewed source.
So as you were saying earlier, there's different methods that these mutations could occur. It could be naturally, or it can be induced through radiation. And two reasons we do this is first to create a more diverse genetic pool, or we're trying to get rid of variants that we don't want or are weaker than other ones. And we use different selective pressures, environmental pressures, such as radiation or chemicals, to get about a desired mutation that we want. We see this naturally in antibiotic resistance. When we use antibiotic on bacteria here, we are killing all the weak bacteria we see here, but what is left is the strong bacteria, and this is an example of mutation selection because only the stronger bacteria is left. Next slide. And as he was saying earlier, a very common method for this is with crops. We see this with a lot of crops that we eat. We use irradiation through conventional radiation we get outside our solar system or on Earth, or we use chemicals that will naturally induce mutations. Crossbreeding too can achieve desired mutations. We may select for different mutations we like or don't like, and we'll crossbreed different plants to have that mutation carry on in generations. And many crops we eat today are through the processes of crossbreed mutations, and they are safe to consume. And earlier, as I was saying, we can take seeds into outer space to use naturally occurring radioactive waves to speed up the evolutionary processes. We can do this on Earth, but it takes a lot longer because the radiation is a lot lower. And once we return to Earth, we can use those naturally mutations that have occurred and continue to create new and new generations and then now we'll have those plants.
So I'm going to talk about plasmids, and plasmids are small circular double-stranded DNA molecules. They are separate from the cell's chromosomal DNA. They can be found in bacteria, archaea, and eukaryotes, but they're mostly found in bacteria. They're not very useful, but they do contain antibiotic-resistant genes, which do help the bacteria survive. So I'm gonna talk about the functions, and again, like I said, for bacteria, they provide antibiotic resistance. And when it comes for scientists, they're useful when introducing new genes to an organisms, and they do this by determining which cells have plasmids, and they insert antibiotic into the bacteria, and those that survive will have plasmids, and those that don't will not have plasmids. And then next, an enzyme will come and cut at the restriction sites right here, and then the gene will leave, and then they can insert a new one. And then, then a different gene can be inserted. This gene is expressed because the promoter site will go over the gene and allow transcription to occur.
Transposons are transposable elements. They're DNA sequences that can move from one location in the genome to another location. They're found in all genomes, so eukaryotic and prokaryotic organisms. They occupy a higher proportion of a species' genome, and they play a significant role in genome evolution, genetic diversity, and genome structure. So the mobilization of TEs, transposons, is called transposition, which is a cut and paste method, and there's also retrotransposition, which is a copy and paste method. So they can positively and negatively impact the genome. The mobilization can promote gene inactivation, modulate gene expression, or induce illegitimate recombination. So in this picture, it shows class 1 and class 2, which is retrotransposition, and just regular transposition. So basically in retrotransposition, it takes donor DNA, and then using transcription, it replicates that portion of DNA into an RNA segment, and then with reverse transcription, it changes into DNA intermediate section, and then it's integrated into the target DNA of the given element. And then in the DNA transposition, which is just the regular, it's basically cut out of the donor DNA and then placed into a target DNA where it's left. And then here are the references we use.